So in the last video, we were talking about osmosis. This is just another way of depicting osmosis using these U-tubes, and they often use these in test bank questions. We will go over these in the Zoom sessions to kind of give you some different problems and different ways that we can look at those. I just wanted to mention that. Okay, so the last couple of ones that are ways that molecules can move in and out of cells. The next one we're going to talk about, number 3, is facilitated diffusion. This is continuing on with the passive types of diffusion. That is, it's a substance that is going down or with a concentration gradient. But in this case, it's a large and or charged substance, and because of that, it has to go through a protein. It still doesn't require energy, but what we're really talking about here is ions, or maybe something like a sugar molecule like glucose or something like that. Now, this is the drawing of the situation. I still have a high concentration on this side and a low concentration on this side, so it's still going to move with a concentration gradient. It just has to go through a protein. Now, there's different kinds of proteins. There's what they call the channel protein, which is kind of this one, where it's just basically the size of the molecule in question. So anything that's that size or smaller would go through, including water, by the way, could go through a channel protein if it's small enough. So anyway, it's going to be going through that channel. Now, another type is what they call gated. I always think of these as like the trash can where you throw the trash on the top and it opens up, and like when you wad a piece of paper, it opens up and lets the paper in and then closes back. This is kind of like that, where the molecule will enter, that will make it open up, and then it will just open right back, accepting the molecules that are on high concentration on the other side. So it basically just reconfigures as the molecule comes in to allow it to go across. So this will be opening and closing spontaneously. All right. Now the next one is active transport. Active transport. This is a active type of movement, meaning that it requires energy, and it is the movement of a large and/or charged substance. And the main reason why it requires energy is that it's going against a gradient, and it is going through a protein. Now there's different ones. Go ahead and write those down. We're going to go over each of those with an overhead because it's easier to draw. I can't draw it really. The sodium potassium pump is hard to draw, so it's co-transport. Over here though, I drawn the hydrogen ion pump just to show you, or so you can have a drawing in your notes if you want it. Here's the phospholipid bilayer, the protein. I have a low concentration on this side and high on this side, and it's going to move against a gradient, requires energy to make that happen. All right, so let's just look real quickly. This is just depicting, this is in your book, depicting the different types. Passive diffusion is going across the membrane directly. Through a protein is facilitated, or if it requires energy, active transport. Okay, so the proton pump. The proton pump is what I just drew for you. This is just showing it going in the opposite direction from what I drew from you, which can happen. It just kind of depends on the situation. Now, this is also called an electrogenic pump. So I just wanted to talk for a second about why that is. If I'm building up charged particles on this side, I'm making what they call membrane potential. That is, if I were to flow a bunch of charged particles through a central point, that actually is an energy creation, just like flowing electrons through a central point will turn on a lamp or something. So electrons, which are charged particles, flowing it through the wire of your the plug in your house that you plug your lamp into that turns your light on, that's just the flow of a charged particle. So in this case, it's a different charge, but it's still a charged particle, and if I were to flow that through a central point, I could make some energy. So I'm creating electrical potential or an electrogenic pump. Okay, The second type is the sodium-potassium pump. Now this is harder to draw. As you can see, it's a little more complicated. So in this case, let's say I'm in an environment like a salty, salt marsh or something like that. And the cell is taking on sodiums, which are ions that might get in there through a facilitated diffusion process, even though they're not specifically trying to get in, or the port wasn't directly for it. It might sneak in. All right, so the cell doesn't like sodium. Sodium ends up being harmful to the cell, so it wants to pump it out. So what it will do is, in a protein, it will load the sodium in the protein, and then it uses ATP to open it up and dump it out. All right, so this is against a gradient, assuming there's more sodium on the outside than there is on the inside, but the cell still wants to get rid of that sodium. All right, now what's going to happen is, because it's open on this side, then potassiums can fit in, and potassiums again might be on a low concentration on the outside, but the cell really uses potassium for many different functions and for osmotic control. And so it's going to want to pump that potassium in. So it's going to take the potassium in and open it back up. All of this was from one ATP. The ATP opened it up. That open space fills with potassium, and then as it goes across the membrane, it's now ready to take on the sodiums again with another little energy bump. All right, so that's the sodium-potassium pump. And the last one I wanted to talk about is something called co-transport. Co-transport uses this electrogenic or proton pump to pump the hydrogen ions across the membrane and create the electrical potential. And that potential in this case is used to bring across a larger molecule like sucrose, which is a disaccharide. Remember, sucrose is like table sugar, but it's also the sugar that is produced in plants during photosynthesis. So the flow of the hydrogen ions back across the membrane with a diffusion gradient through this protein will pull that sucrose with it, or basically it provides the energy for bringing that sucrose across the membrane. So this is called co-transport.